In Lecture 2, we want to discuss the origins of the pH scale and look at some tools that will help us to understand the chemistry of acids and bases a little better. If you recall from Lecture 1, water is an amphiprotic molecule. It's capable of acting as either an acid, an H plus donor, or a base, an H plus acceptor. Water can act as a Bronsted acid in its reaction with aqueous ammonia. Here, water is acting as an H plus donor to ammonia to form ammonium ion and hydroxide. On the other hand, water can act as a Bronsted base, in this case, acting as an H plus acceptor from acetic acid to form hydronium ion and an acetate ion. The amphiprotic nature of water means that the following reaction is possible. One water molecule could react with another water molecule, and if one of them acts as an H plus donor and the other as an H plus acceptor, you would end up with a hydronium ion and a hydroxide ion from these two water molecules. Now we could also write this as 2H2O in equilibrium with a hydronium ion and a hydroxide ion. Or if you prefer, we could write it as water ionizing into hydrogen ion and hydroxide ion. Now in pure water, this autoionization, meaning automatic ionization or water ionizing by itself, occurs to only a very limited extent, and the equilibrium constant for the process is quite small. We could write an equilibrium constant expression for the reaction written like this. We'd have the concentration of hydronium ion and hydroxide, each raised to the first power, divided by the concentration of water squared. We remember, of course, that water liquid does not appear in the equilibrium constant expression, and so we would just have a hydronium ion concentration times hydroxide. It turns out that the equilibrium constant for this, as I said, is quite small. It's 1 times 10 to the minus 14 at 25 degrees C. And we have a little note here that came in early that the equilibrium constant for this autoionization is usually labeled with a W for water. So it's the autoionization constant for water at 25 degrees C. Now this is incredibly small for an equilibrium constant, which means, of course, that the equilibrium strongly favors the reactant side. So if we look at the reaction we wrote this from, we're saying that this equilibrium strongly favors the side of water molecules, not the side with hydronium and hydroxide. And this is quite good news for us, because if this were not the case and the equilibrium constant were much larger, water would spontaneously degrade into hydronium ions and hydroxide ions, and we would cease to exist. So it's a good thing that this equilibrium constant is so small. Now, even though the value of that equilibrium constant, the water ionization constant, is really small, it's still possible to calculate the tiny concentrations of hydronium ion and hydroxide ions that are produced in pure water. To do this, we can simply set up an ice table and calculate the hydronium ion and hydroxide ion concentrations as for any equilibrium. So let's do that. What I've done here is set up an ice table. We have, of course, water on the left, hydronium ion and hydroxide on the right, and we're going to assume the concentration of hydronium and hydroxide ions start out at 0. Recall that in an ice table for a liquid or a solid, we don't deal with that in the ice table. We just put a dash in there. So I'll complete the ice table for water liquid, and we simply have hydronium ion and hydroxide ion changing by some value x. Of course, they both be plus x. And quite simply, at equilibrium, we have a concentration of x for the hydronium ion and x for the hydroxide ion. You can see that whatever x is, we're going to end up figuring out that in pure water, the concentration of hydronium ion and hydroxide ion will be equal. But let's see what the value is equal to. If we went ahead and wrote the equilibrium constant expression and then substituted in for the hydronium ion and the hydroxide ion concentrations at equilibrium, which we said were each x, we'd have x times x, which is x squared, and we simply set it equal to the value of the autoionization constant for water. So you can see solving for x here is fairly straightforward. We just take the square root of both sides, and we find out that x is equal to 1 times 10 to the minus 7. Well, what does that mean? That means in pure water, the concentration of hydronium ion would be 1 times 10 to the minus 7 molar, and the hydroxide ion concentration would be equal to that, 1 times 10 to the minus 7 molar. They're both extremely small concentrations. One could say ridiculously small concentrations, but the concentrations are equal for both of them. So we would say something that you already know. Pure water is neutral because the concentration of hydronium ion, the acidic ion in water, is equal to the concentration of hydroxide, the basic ion in water. Now, in practical terms, pure water is a challenge to produce. For example, a small amount of carbon dioxide from the air easily dissolves in water and produces the weak acid carbonic acid that ends up increasing the concentration of hydronium ion and ultimately, as we'll find out, decreasing the concentration of hydroxide. But nevertheless, in pure water, the hydronium ion concentration is 1 times 10 to the minus 7 molar, and the hydroxide ion concentration is 1 times 10 to the minus 7 molar. Now, in any aqueous solution, even if there are many other molecules and ions present in that solution, the following is always true at 25 degrees C. And I have this ridiculous figure come down that says, attention, this is really important. Well, what's always true? It's always true that if you take the hydronium ion concentration and you multiply it by the hydroxide ion concentration in that solution, it will always be equal to 1 times 10 to the minus 14. Or, if you prefer to write hydronium ion as H+, the H+ concentration times hydroxide is always equal to 1 times 10 to the minus 14. Interestingly, this holds in any aqueous solution. So it doesn't matter if there's many other things dissolved, like in a lake or a pond or a beaker of weird-looking aqueous solution in the lab. The hydronium ion concentration times hydroxide will always equal 1 times 10 to the minus 14 at 25 degrees C. Now what this means, of course, is that since the product of these two is a constant, that there's an inverse relationship between the concentration of hydronium ion and hydroxide. And what we mean by that is that if by chance the hydronium ion concentration goes up, like you add an acid, well then by necessity the hydroxide ion concentration would have to go down so that the product of these two comes out to be 1 times 10 to the minus 14. If you did something to increase the concentration of hydroxide, then the hydronium ion concentration would have to drop in order for the product of the two to always equal 1 times 10 to the minus 14. So for example, suppose an aqueous solution was found to contain a concentration of hydronium ions equal to 4.66 times 10 to the minus 4 molar. What would be the concentration of hydroxide ions in this solution? Well, we'll write hydronium ion concentration equals 4.66 times 10 to the minus 4 molar. We don't know what the hydroxide ion concentration is. However, we do know this relationship is always true. The hydronium ion concentration times the hydroxide has to come out to be 1 times 10 to the minus 14. So what we'll do is we'll simply solve for the hydroxide ion concentration. We'll bring the hydronium ion concentration underneath the 1 times 10 to the minus 14 right here and just substitute in the given hydronium ion concentration. Do a little math. We find out that the hydroxide ion concentration is 2.1 times 10 to the minus 11 molar. So in this solution, the hydronium ion concentration is 4.66 times 10 to the minus 4 molar. The hydroxide ion concentration is 2.1 times 10 to the minus 11 molar. As we said before, if the hydronium ion concentration goes up, gets a little higher, then the hydroxide ion concentration would have to drop. So a couple things to note about the solution in this example. First of all, this solution would be considered acidic because the hydronium ion concentration is greater than the hydroxide ion concentration. 4.66 times 10 to the minus 4 is a much larger number than 2.1 times 10 to the minus 11. The other thing is that it's about time we defined pH, and we're going to say that the pH of any aqueous solution is based on the negative of the base 10 logarithm of the hydronium ion concentration. In other words, simply pH equals minus the log of the hydronium ion concentration. Or if you're writing hydronium ion concentration as H+, pH is minus the log of the H+ concentration. For this particular solution, pH is minus the log of the hydronium ion concentration, which would be minus the log of 4.66 times 10 to the minus 4. Now you should note here that 4.66 times 10 to the minus 4, the log of that gives a negative number, but we're taking the negative of that negative and we end up with a positive. So the pH is 3.332. And that makes perfect sense from what you'd remember from high school chemistry or even junior high physical science courses. The pH is less than 7, so the solution's acidic. And we already said that because the hydronium ion concentration was greater than the hydroxide ion concentration. Let's more formally introduce the pH scale and a few other things on the next slide. Again, since we're using base 10 logarithms, we can say the acidic or basic character of aqueous solutions is typically tracked as powers of 10 of the hydronium ion concentration, or in other words, base 10 logarithms. So here's a chart that I just suddenly threw at you, and let's see what's going on here. Let's look at the middle column that's highlighted here, the hydronium ion concentration. You'll notice that we're giving concentrations in powers of 10. 1 times 10 to the minus 14. 1 times 10 to the minus 13 is 10 times bigger. 1 times 10 to the minus 12 would be 10 times bigger again. It doesn't mean the hydronium ion concentration can't be numbers between these, but I'm just giving powers of 10 all the way down to 1. Curiously, and we'll get back to this in a second, but you'll notice that a pH of 14 corresponds with a hydronium ion concentration of 1 times 10 to the minus 14. A pH of 13 corresponds with a hydronium ion concentration of 1 times 10 to the minus 13. So a drop of a factor of 10 shows up in the pH scale as a difference of 1. It's really a difference of a factor of 10 in hydronium ion concentration. Now, right next to the hydronium ion concentration is the hydroxide ion concentration. And you'll notice in every double entry in this table for hydronium and hydroxide, if you take the hydronium ion concentration and multiply it by the hydroxide, it always comes out to be 1 times 10 to the minus 14. Example, 1 times 10 to the minus 14 times 1 is 1 times 10 to the minus 14. Let's go down to pH 8 here. 1 times 10 to the minus 8 times 1 times 10 to the minus 6 is 1 times 10 to the minus 14. So you notice as the hydronium ion concentration gets bigger, the hydroxide ion concentration gets smaller, and the product of those two is always 1 times 10 to the minus 14. Now on this side, we've listed the pHs, and the pH is equal to minus the log of the hydronium ion concentration. We pointed this out before. You'll see that these exponents here are always equal to the pH because of this relationship here. Now, when the hydronium ion concentration and hydroxide ion concentrations are equal, the solution's neutral. That would be a pH 7 because minus the log of 1 times 10 to the minus 7 would be 7. That would be neutral when the hydronium ion and hydroxide ion concentrations are equal. Down here, where you know the pH has gone acidic, less than 7, you'll see that in all of these cases, the hydronium ion concentration is greater than the hydroxide ion concentration in all these area in here in the red. If the pH is greater than 7, you know that's basic, and this is where the hydronium ion concentration is less than the hydroxide ion concentration, and so the solution would be basic. So the pH scale is based on the negative log of the hydronium ion concentration. Well, interestingly, we could do the same kind of math on the hydroxide ion concentration and calculate the pOH. The pOH would be minus the log of the hydroxide ion concentration. So by now you should notice that this small p is a symbol that means minus the log of. P minus the log of. So if we took minus the log of the hydroxide ion concentration, we'd get a pOH. And for a hydroxide ion concentration of 1, the pOH would be 0. And they'd go 0 to 14. And you'd have all the same ranges. The pOH is 7 when the solution's neutral. That's when the hydroxide ion concentration equals the hydronium ion concentration. The solution is acidic when the hydronium ion concentration is greater than hydroxide. Interestingly, this would be when the pOH was greater than 7. And the solution is basic when the hydronium ion concentration is less than hydroxide ion concentration. And in this area, the pOH is less than 7. You'll notice that in every case, the POH plus or the pH plus the POH always adds up to 14. And as a corollary here, the hydronium ion concentration times the hydroxide always equals 1 times 10 to the minus 14, but the pH plus the POH equals 14 in all these cases. Now, almost all the time, we will talk about pH, which means we're going to be thinking about the hydronium ion concentration and whether it's greater than or less than the hydroxide ion concentration. However, knowing the definition of POH will give us some alternate ways and sometimes maybe some shortcuts in some calculations involving acids and bases. So let's look at a few example problems where we'll talk about pH and possibly POH, so forth. So here's one. What would be the pH of a solution where the hydroxide ion concentration is 7.24 times 10 to the minus 9 molar? Well, one way to approach this is as follows. We know that the hydronium ion times the hydroxide ion concentrations equal 1 times 10 to the minus 14. We're given the hydroxide ion concentration, so let's just solve for the hydronium ion concentration. So here we go. We're solving for hydronium ion concentration, and we find out that it's equal to 1.38 times 10 to the minus 6 molar. So just a note here, we're carrying extra sig fig for a minute. We know that the pH is minus the log of the hydronium ion concentration, which we just calculated. So pH equals minus the log of the 1.38 times 10 to the minus 6, and we get a pH of 5.86. It's acidic. So we know this solution is acidic for two reasons. One, of course, is because the pH came out less than 7, but also because we calculated right at first that the hydronium ion concentration was greater than the hydroxide ion concentration. Now, a word about significant figures here. You might remember from high school math that since 1.38 times 10 to the minus 6 has 3 significant figures, and that's a little bit of a lie because we carried an extra one here, but let's see this through. The number of decimal places in the pH should be 3, not 2 as shown. So truthfully, we did carry an extra sig fig from the previous calculation. We said the hydronium ion concentration was 1.38 times 10 to the minus 6 instead of 1.4 if we had rounded off to the correct number of significant figures. So if we remember that there really should only be a 1.4 there, in other words 2 sig figs, then the logarithm would have 2 significant figures beyond this number 5. If you recall, this is the 0.86 would be the mantissa of the log, 5 the characteristic. Well, if this is all very confusing, one thing that'll clear this up is that often, no matter how many significant figures are shown here, we'll often see pH written with 2 digits beyond the decimal point. And the reason is because pH meters are usually only good to about the hundredths place anyway. And when I say no matter how many significant figures here, what I'm meaning is if there were many significant figures here, you might still see only 2 places beyond the decimal point for a pH because that's pretty much the precision of most pH meters. Now, another way to approach this example would be to calculate the POH from the given hydroxide ion concentration. So let's do that and see what happens. POH would be minus the log of the hydroxide ion concentration, and we'd get 8.140. Notice there are 3 significant figures here, so I am going to carry 3 places here for just a minute. And we'd remember that pH plus pOH always equals 14, so we could just solve for pH, and we'd get 14 minus 8.140, and we'd get, following significant figure rules, a pH of 5.86, which agrees with what we got by this previous method. How about this example? What would be the hydronium ion concentration and the hydroxide ion concentration in a solution with a measured pH of 8.77? Well, we do know something right off the bat here. If the pH is greater than 7, we know this is a basic solution, so we know the hydroxide ion concentration is going to end up being greater than the hydronium ion concentration, but we want to know what the concentrations are exactly. So one way to approach this is to write our definition of pH, and then we'll put our known pH in, 8.77, which is equal to minus the log of the hydronium ion concentration. And now we'll do a little math. What I'm going to do is multiply both sides by minus 1, which is effectively like just bringing the minus over to the other side. So we have minus 8.77 equals log of the hydronium ion concentration. And then what we're going to do is take the anti-log of both sides. And simply, that just means we're going to take this side and use it as an exponent on 10. So we're going to take 10 to the minus 8.77. If we do that to the left, we've got to do this to the right. So we take 10 to the log of the hydronium ion concentration. This is effectively taking an anti-log of both sides. Well, we can figure out what 10 to the minus 8.77 power is by just using a calculator. And you find out that it's equal to 1.7 times 10 to the minus 9. Well, what about this mess on the right? Well, 10 to the log of the hydronium ion concentration, where the 10 and the log effectively cancel each other, and we're just left with a hydronium ion concentration, which was what we were after. I suppose I should have put molar there, but I didn't. But of course, the brackets here imply molar concentration. Now we can remember that the hydronium ion concentration times a hydroxide is always equal to 1 times 10 to the minus 14. So we'll solve for the hydroxide ion concentration using the hydronium ion concentration that we got from the pH. And we get a hydroxide ion concentration of 5.9 times 10 to the minus 6 molar. Well, here are some things to note here. Remember, the hydronium ion concentration times hydroxide always equals 1 times 10 to the minus 14. I hope by this point you're getting sick of hearing me say it. That's good. If I put the numbers that we got for our hydronium and hydroxide ion concentrations, you'll notice that's true. And remember, the pH plus the pOH always equals 14. Well, the pH was 8.77. The pOH would be minus the log of the hydroxide ion concentration, which would be minus the log of our 5.9 times 10 to the minus 6. We get 5.23. And yes, 8.77 plus 5.23 equals 14. Now, can we measure the pH of a solution in the lab? And of course, you know the answer to this is yes, and most of you, I'm sure, have done this. You could use a pH meter. Well, what's going on with this pH meter here that you can see is reading 7, and here's our solution, and we're using some sort of pH electrode. The pH electrode, it might be a little hard to see, is actually an electrode that's made out of glass. And what this does is this glass electrode is hollow and it has a solution in it. And so the glass electrode measures a potential difference in millivolts between an internal reference solution inside the glass electrode and an external solution, which you're measuring, and converts this voltage to a pH value. There's two electrodes here, and the additional electrode in this picture is just a temperature probe, and the meter is reading the solution is having a pH of 7 at 25 degrees C. Now, pH meters must be properly calibrated before use, so if you're in a lab, you'll probably use what's called some standard buffers to calibrate the pH meter. And also, it's common for the bottom of the glass electrode, which is like a little glass bulb, is covered with a plastic cage because that glass bulb is very thin glass and you want to protect it. The other way that you could measure pH is in a less precise way, but you can use organic dyes that change color over particular pH ranges. So here's an interesting picture from another general chemistry textbook that we are not using in this course by Daryl Ebbing and others. And what we have here are different organic dyes and what color they are at different pHs. So for example, a common dye that's used in high school and junior high labs is phenolphthalein, which actually stays colorless in acid solution and actually at pH 7 and 8, but suddenly turns to a dark pink color at pH 9 and above. Bromethymol blue is an interesting dye. It stays yellowish in acid solution, but it gradually turns blue between about pH 6 and 8. So you'd want, depending on what pH changes you're expecting, you might make a wise choice of which indicator you wanted to use. So, for example, suppose you were interested in monitoring a pH change between 3 and 5, then bromethymol blue wouldn't be a great choice because it stays yellow between 3 and 5. However, methyl orange down here turns from red to orange over that range, although it might be hard to detect the actual change from red to orange. Finally, an indicator that you see often in general chemistry labs is a universal indicator, which is actually a mixture of organic dyes. And generally speaking, the colors are red at low pH, oranges at a little higher pH, yellow around pH 6, green at pH 7, and then blue, purple, and this very dark purple at higher pHs. An interesting thing about this figure is that I don't recall universal indicator being orange at pH 1 and 2. From my experience in the lab, below about pH 4 or 3, it just stays red and then turns orange and then a little higher orange and then yellow. So these are two ways that pH can be measured.